Okay, so I was given the task of considering what's the difference between secretomes and exosomes. And when I looked at this originally, I decided to give my version. And then because I was practicing Grok 3, because it was part of the development of Grok 1 and 2, I threw it at it to see what its consideration was. So we know this sort of regenerative medicine is one of the big things coming our direction now. So this was mine. If a human organism was an orchestra, the secretome would be the orchestra itself. Growth factors would be the baton, regulating cellular behavior with specific things. But exosomes would be related to a violin solo, a moment of spotlight amidst the tapestry of depth and richness of the performance. That was mine. So I threw it out to Grok to see what it would think. And it gave me back these sort of visualizations, which is very interesting. They said they would consider it couriers with sealed letters. So in other words, almost like the violin solo being an individual instance in the midst of the whole orchestra, they're seen it as well-dressed couriers on bicycles or scooters weaving through the streets with a specific letter going to a specific house. So you have to have almost a specific address. It's not like a wild message to everybody. And the letters also would glow faintly, and that'd be hinting at the precious cargo meant only for the recipient, like gene-regulating RNA. So it's an interesting analogy. So I threw it up then what it would think of secretomes, and they said secretomes are like the billboards in a city. Somewhere it looks like a bit like Chicago, that display bold messages, grow or alert. It's radio towers transmitting messages to sort of everybody, broadcasting onto the radio stations, or the mail trucks below that are general delivery. So it's not like some of our delivery systems that specifically come to one address. So I thought that was a sort of cute analogy. So exosomes, as we know, look like this. They're vesicles that are extracellular, that are made by all eukaryotic cells. They vary in size from 30 to 150 nanometers, and they contain a variety of biomolecules, including mRNA. So this is important. And mRNA and other, I suppose, non-coding RNAs. So the thing we must understand with all these is signaling. The whole secret in cells and what's happening is the ability to understand signaling. So they're surrounded by a lipid membrane, a bit like, I suppose, during COVID when the mRNA vaccines come out. It's almost the same type of technology. In other words, the vesicles come up to a cell, they bind with a receptor, and they get into it that way, where secretomes are often intracellular, so they're the sort of mass product within the cell that also contain exosomes per se. So without, at the risk of getting political to an extent, I know one of my earlier lectures used analogies that way as well. So exosomes are, I suppose, like targeted missiles that are sent from one area specifically to hit another area, where secretomes are not like that at all. They're more broader, and they're almost like a bomb that will go off in an area and just blow everything. So secretomes per se are a totality of all the different messengers. They play an essential role in those three big things: cell communication, tissue repair, and immune response. And in my last lecture, I know some people are here that were at it, I compared it, I suppose, to an extent to American politics, but not really getting involved in politics. These things are the same as we have in the world around us. They're just a sort of microsomal thing within a cell. So if we look at the Republican Party and the Democratic Party at the moment, the infrastructure that sort of America needed was proposed by the sort of Democrats, and to an extent, this is the same as tissue repair. The communication within society, in other words, what we should be mentioning within our schools and what should be learned, whether it's sort of creative theory or not, you know, this is communication within cells we have the same thing this way. And their immune response, of course, is defense. So we say the Republicans want to take over Greenland as a defense for America stroke the rest of the Western world. Immune system is like that. So we have the same analogies. These, what all these things are doing, one of those three things: communication, immune response, or wound repair. And in society, we have exactly the same things. So if we look at secretomes per se, they'll come from MSCs, mesenchymal stem cells. And we've been using the equivalent in a lot of award-winning projects that I've done, like the PLUS theory and sort of Helper, where in terms of, I suppose, using stem cells, we were doing that now almost, 7, 8, 10 years ago, maybe more. Okay, so secretomes is an umbrella term which includes lipids, anti-inflammatories. They also contain the metabolites, fatty acids. They also contain proteins that facilitate cell-to-cell communication. And they also obviously contain exosomes as part of their cargo, per se. So they're almost like a generalized one. So again, just like exosomes, they're involved in tissue and hair repair. They can restore the integral structure of damaged tissues, as we would have done a lot with, I suppose, impending necrosis with vascular occlusion. We had a lot of cases that we had to bring back from 10, 14 days that we brought back successfully by using the same type of technology, bioregenerative, particularly through stem cells at the time. Now, we have to understand, I suppose, biochemical cues. That's as important as signaling. I think if we understand signaling, then we sort of can understand how all these different things work. In terms of signaling, there's a couple of different things. There's autocrine, there's endocrine, and there's paracrine. So we look at them through a little sort of analogy that I have here.
To make a multicellular organism, cells must be able to communicate with one another. And to do it, cells often send out tiny chemical signals that act on the receptors of other cells. Signals can be classified according to the distance between the signaling cell and the target cell. Autocrine signals are produced by a cell and go to its own receptors, so the cell sends a signal to itself. Paracrine signals are produced by a cell and go to target cells that are nearby. And endocrine signals are produced by a cell and go to target cells that are further away.
Hormones, don't forget.
Examples of these include hormones that are secreted into the bloodstream, as well as cytokines that can be released at the site of injury and act on the brain to cause a fever. Signaling molecules, or ligands, can be hydrophobic, meaning that they tend to repel water,
this is
or
why some of them need
hydrophilic, meaning that they tend to stay in water.
And some of them
Hydrophobic signaling molecules can't freely float in the extracellular space, so they're brought into the target cells by carrier proteins. Hydrophobic molecules can diffuse across the cell membrane and bind to receptor proteins inside the target cell, either in the cytoplasm or in the nucleus. Most signal molecules are hydrophilic, so they can freely float in the extracellular space to reach the target cells, but are then unable to cross the cell membrane. So to pass on the signal, hydrophilic molecules bind in receptors on the cell surface. These receptors are transmembrane proteins with an extracellular end that binds to the ligand, and an intracellular end that triggers a signaling pathway inside the cell.
And I suppose that was so important during COVID when we noted originally,
the first stage is
we don't have to watch that, COVID could only get into the cell through a receptor, which at the time was the ACE receptors. And when we started having problems, we thought we would use different things like lisinopril to stop the nodule formation through angiotensin converting enzyme type 2. So again, they're all doing one of these three things: repair, communication, or defense. And as I said earlier, that's equivalent to what we see in the real world as well. So we could see almost the difference between them also, as a postman is almost like exosomes. He's coming with a parcel of letters, but each one of them is going to a specific area. The train driver is different. He's bringing a lot of people as his cargo, but they're all getting off and going to different addresses. And the little delivery boy, I suppose, is self-explanatory. So all the Qs refer to growth, differentiation, or function, and they'll use hormones, extracellular matrix components, as Diana was alluding to earlier, and Neve was also, and cytokines. And it's when you mention in your lecture, Neve, I've just seen the calcium hydroxyapatite, the ceramic versus the PLA, I suppose, as a foreign body. I'm astounded always to think when we're using PLA, and we're using it now for 25 years, that we see it every day in plastic mugs. That's what actually those little containers are, the PLA sort of polymers, you know. So how they don't sort of cause more problems is amazing, but I get great results with sculpture. I like it. So again, what is the similarity and what's the difference? The similarity, as we mentioned earlier, is they both contain signaling molecules. And if you understand, I suppose, how signaling molecules work, you can see the similarity between them. They both will modulate cell behavior, and they both contain a diverse array of biomolecules. So the difference between them is obviously the mode of release. Exosomes are derived from endosynthetic pathways within cells, whereas secretomes originate from a secretory pathway. In terms of structural composition, the difference between them is in the supply of nutrients, how they facilitate intracellular communication, support detoxification processes, and how they regulate immune responses. Again, this is me playing with AI. I loved your AI models, actually. I seen them immediately, and I said, that's what I was doing also. So they compare to a broad collection of bioactive molecules secreted by cells. Both of them play a role in cellular communication, tissue repair, and immune response. And secretomes are normally derived from stem cells, such as mesenchymal stem cells. And we switched over, and I'm not doing product promotion here, but from using stromal factors when we used a lot of Vaser to calcium from Singapore, but they're from New Zealand, red deers. And if anybody hasn't used it, I'm telling you, the results we got, we did comparative studies later with both those and exosomes as a split case with hair growth, and I still think they outperform it. I know they contain exosomes as well, but this is going to be a market that we're all going to have to live with and learn with. And it's interesting, the young generation of doctors coming in are totally focused on this, and behavior has changed in terms of dermal fillers or bioregenerative, but it's a new age, and really, as yet, I think, without sort of having a dig at China, 10 years ago, we were getting a lot of poor products coming as cargo, and I'm in out of China a lot. They've given me a big award recently, so I love the place. So as a consequence, the products now are so good that are coming, and it's almost the same thing. We're at where China was 10 years ago. We've got products, but the cargo is poor quality. It's only as good as the person that is getting it from, and it's not even age related. Some people can regenerate much better than others. So until we learn to control the cargo and what goes into it, we'll not be masters of our own destiny. And as I said that as well about controlling telomeres, which is going to be the new one in terms of anti-aging. If you have got short telomeres leading to cell senescence, okay, if we control telomeres, we know we can get long ones and we live longer. But also, if there's cancer on board, you have a good chance you'll get it and die from it. So it's the control of all that that really will be the important thing. So I'm down to a minute. Let's see what we've left. Okay, there's a little quick difference between the two of them. In terms of definition, secretomes are a collection secreted by the cells, whereas exosomes are small vesicles carrying specific molecules going to a specific address. The size of exosomes is limited to 30 to 150. It's not limited to vesicles in terms of secretomes. The mode of action are sort of similar, but if we can control the cargo of exosomes, then we'll get a much better result of what goes into it.
A minute.
Okay, thank you for that. Secretomes have broad applications, whereas exosomes are usually targeted regenerative response. And I think that's me. Smack on time. And then the next thing, of course, will be cellular aging, telomeres, cell senescence, and particularly secretory phenotype senescence-associated proteins. There we go. Thanks.